main layers in the wall of the eye. What is this main layer from here to here?
The retina.
Yeah, it could be a retina, or you could say neural tunic, same thing. How about this main layer here to there?
Sclera.
You could do sclera. Yeah, actually, I guess that's the only right answer for that, because if you say fibrous tunic, that also includes the cornea, so it's got to just be sclera. And then main layer from here to here?
Choroid.
The choroid. Good. Okay.
The innermost is the cornea.
Hmm?
The innermost one is the cornea.
The innermost one is the retina.
A little
Oh,
closer.
retina. Okay, retina.
Good today. Alright, so now in this main layer we have two sort of main sublayers. This one over here kind of looks a little brown. It's a little kind of scattered in this slide. The other slide you saw was better. But what is this
That's the
sublayer right here? That's of the retina.
pigmented of the retina.
Yeah, it's just pigmented layer, or you could say pigmented epithelium. What does it say in here? It says pigmented layer, or pigmented sublayer. Okay, and then all the rest of these are well, I guess there's multiple sublayers. So then let's identify the cells. So these are nuclei of what kind of cells?
Rod. Okay. Glias. Or.
Wheel? Is that the ganglion?
Rods and
Not
cones?
the ganglion. You got three choices: bipolar, ganglion, or rods and cones.
Rods and cones.
Bipolar. Yeah.
I thought the bipolar was no, that's the glia cells. That's one.
So it goes. This is the choroid. Okay, then you have rods and cones here, and then you have bipolar neurons or cells, and then ganglion. Okay, so the light's coming from this way. This is outer. Sclera is here. And choroid is here. Okay, so then you have the light coming in this way. It's got to go, goes past these other cells, and it's a little counterintuitive, but the rods and cones are the ones that are stimulated by the light, and then they relay the messages back this way. Ganglion cells carry it all the way back to the optic disc. Does that make sense? Yeah? That makes sense? You may or may not be able to see it. Can you see it? Can you turn the light on? Deborah, can you see over here? Or no? No? I'll turn on the light for a minute. You can't have everything, huh? Either remote control light. There. So it's just looking at the layers of the eye. Okay, so outer is out here. Sclera. It's represented by my horizontal lines. Choroid is the middle one. Here represented by the dark. Colored. And then, of course, we have the pigmented layer. That's right in here. I didn't put that in. Okay, and then you have all your cells. So rods and cones are the innermost, then bipolar, and then ganglion cells. And the way to identify them easiest is to look for the nuclei, because they're connecting to one another. So where one ends in here and the other begins, it's hard to tell. So that's why I reference the nuclei. Okay, they show up well, and we know that belongs to those cells. Okay, so innermost is ganglion cells, bipolar cells next, and rods and cones next. That's the way to remember it. Okay, the scattering of the cells, those are the ganglion cells. That's another sign. Those ones. These two layers, they look pretty similar. Sometimes this one looks bigger than that. Sometimes it looks bigger than this one looks bigger. Okay, so it's just positioning. Okay, so then light comes in. I don't know what that is. Light's coming in. Well, that's epithelia. That's what you're going to learn tonight. That's simple columnar epithelium. And so light's coming in from this way. Okay, right? These are your receptor cells, the rods and cones photoreceptors. They're stimulated by that. They send that electrical signal to the bipolar cells, who then communicate it to the ganglion cells. And then ganglion cells are going to carry that, where you have all of the axons synapsing with your next neuron right here at the optic disc, where you have the blind spot. Okay, so let's go back to this then. So then, once again, these are the nuclei of bipolar cells. Good. These are the nuclei of what kind of ganglion cells. Okay, what main layer is this? Sclera. And these are nuclei of what cells?
Rods and cones.
Rods and cones. Good. And this main layer is?
Choroid.
Choroid. And what about this small sublayer?
The pigmented.
Pigmented
Layer
layer.
of the retina.
Very good. Okay, so that's good with this slide. So now we need the one where we can see the whole eye. All right, this one's F23R, in case you want to reference your lab or look back at it later in the library or during class. Monty, will you do the light back down? I think that will be better for us. Down, down, up, down from the left.
Yeah. Is that the one that you wrote on the board the other day?
Yeah, this is the one that we added that was actually superior F20 to F22. This is F23 is superior. Although this one doesn't have as good an optic disc as the other one. Okay. So here you can see, you should be able to look up there and recognize that same organization of the wall of the eye, right, with the three main layers. Can you see it up there? Yeah?
Yeah.
Okay. Okay, so that's what's sort of normal, right? And then we move into this area. We're at the posterior part of the eye. So I might give you a hint as to that. Here at the posterior part of the eye. Okay, anterior is over here at this end. Okay, so you see how the organization, it totally changes and disappears right here. See that?
Mm-hmm.
Yeah, okay, so then you can see the whole thing. The way it stretches from here to there, and then if we go this way, we see this
optic nerve.
This same structure, well, related structures coming out the back. Yeah, so this structure, what's this structure?
Optic nerve.
That's the optic nerve. Okay, that's the optic nerve. That's coming out of the eye, organized nerve of what? What's in there? What's inside there? Somas or axons?
There's
What's inside of that nerve?
nerves or
Somas or axons? Axons. That's right. Nerves carry axons. Good. It's a hard concept. Okay, and then, so then this here, this structure reaches all the way to the inside. It's the other side of the coin. Okay, so if you have heads on this side, you got tails on this side. The optic nerve is the heads, the tails is
the optic disc.
That's the optic disc. Okay, that's the optic disc. That structure is the optic disc. Okay, that was the structure then that we did the cow eye. You were supposed to probe the retina, the white flimsy part, and I was trying to get you to kind of hang the retina off, and then you can see how it all gathered and was connected back at the optic disc. Okay, so let's go anteriorly. Lots of little dust particles. Some of you even cut open the lens, and you were able to see how there's concentric layers of collagen fibers. I think it was Christy, yeah, and Deborah. That was pretty cool. Okay, so what's this structure here?
Lens.
That's the lens. Okay, big oval shaped structure. You can go down the other end of it. There it is. Okay.
That little strand, the suspensory
There
ligament.
might be a little strand of it. Let's see.
Guess not.
No, probably not. A little. Yeah, I don't know. Almost looks like a piece of the lens that kind of unraveled. Although I guess it could be. I've never seen a suspensory ligaments on the slides before. So here is the anterior eye. Let's get a little closer to that. That's the anterior edge of it. Yeah, so that is the cornea. That's right. So this structure from here to here is the cornea. Okay, so then what is this very thin layer? It's a squamous layer.
The non-character.
Yeah, it's the conjunctiva, conjunctiva, or however way you want to say it. And it's the ocular one. Remember I added that one to your lab. It just said conjunctiva, but I want you to specify ocular. Okay, so there you can see the non-keratinized stratified squamous layer. The ocular conjunctiva. Okay, and there's a space. If we move posteriorly, there's a space located between the cornea and the lens, and it also includes the space down. Includes the space down in here. What is this?
Anterior chamber.
Not chamber. Chamber has slightly different meaning.
Anterior cavity?
This is the anterior cavity. Includes both of these. In your book, it divides it up into chambers.
Yeah, that's why I was getting
I didn't
confused.
cover that. So if I just pointed to the front and you said anterior chamber, you're technically correct, and that would be fine. I didn't cover chambers, so we just did this whole thing as the anterior cavity because it's all the same thing anyway. It's all aqueous fluid, aqueous humor. So if you're correct with the chamber, that's fine. Otherwise, don't worry about the chambers. Okay, so anterior cavity, yes. Anterior cavity, anterior cavity. Okay, and then on the posterior side of the lens, there's quite a large space. And so what would this space is called?
Posterior cavity.
Yeah, posterior cavity. Okay, and then we did the cornea, the ocular conjunctiva right there. We did the anterior cavity. We move posteriorly. We have this pigmented structure.
Iris.
Yeah, the iris. So here's the iris. This is the smooth muscle, if you're wondering, and the pigmentation is over here. And then there's an opening where the iris stops, and then it continues on the other side. Real little tricky to just point to it and ask what is it. So I kind of have to frame the question, correctly so that you say pupil and not anterior cavity. But yeah, I could say something like, well, what is this opening that this structure controls the size of? So it would be, what is that? The pupil. Yeah. So the iris controls the size of the pupil. So when you say pupil dilates or pupil constricts, that's what the iris, the actions of the iris, the muscles in the iris that are causing that. Okay, so there's the other side of the iris. Here. out of the three tunics, the fibrous outer tunic, the vascular tunic, and the neural tunic, which one is the iris part of? Is it part of the outer fibrous? Is it part of the vascular tunic, that's the middle one, or is it part of the inner neural? Yeah, it's part of the vascular one. That can be a little tricky to remember. If you look at it like this, you can see it's a continuation, really, of the choroid. Remember, vascular tunic is choroid, ciliary body, and iris. So the answer was vascular tunic. So this is the choroid. It's just a little sparse right here. It's kind of spread out a little bit, but it's got a lot of pigmentation. Dark. You follow it this way. Okay, and here the smooth muscle starts to come in. That's part of the ciliary body, and you follow it up. Okay, so then this is the ciliary body. So if you follow the choroid, it changes to smooth muscle and it kind of thickens. This is called the ciliary body. These little processes are also part of that, but were those on your list?
Yeah, both were on.
The ciliary processes were on there. This is really good. We didn't see ciliary muscle. Okay, yeah. So this entire structure is the ciliary body. See the smooth muscle along with these. These are the ciliary processes. This is where the suspensory ligaments would attach down here to the lens. See where you are? So here's the lens. I know it's hard on this.
So are they destroyed in the process?
They've been destroyed, yeah. Okay, so then what is this entire structure here?
Ciliary body.
That's the ciliary body. Good. And how about just these?
Ciliary processes.
Those are the ciliary processes. So ciliary processes here, ciliary body includes the processes. Yeah, I know this is the ciliary muscle in there as well. Okay, so you follow back up. You see it changing over to which main layer?
Vascular.
Yeah, that's the vascular tunic, but more specifically,
Choroid?
the choroid, yeah. Okay, and it goes back to that kind of a mess over there. Here it's a little better. See, so you follow it all the way from the ciliary body. Now it looks like your more typical looking choroid, the dark on the sides. And it has lots of blood vessels. That's why it's called the vascular tunic. Any questions? Any other parts you want to see or that I missed?
Are we supposed to know the difference between the ocular and the palm?
Oh, the palpebral. Yeah, I did write that on the board. Thank you. Yeah, there it is on the list. Yep. I was going to remember it. That little hair or something on there. There it is. Okay. That's pretty weird, huh? The eyelid. No eyelashes, though. Cows have eyelashes?
Do cows? I think they
Yeah,
do.
I think
They do, definitely.
Yeah.
They removed them.
They looked in the mirror and plucked it with their tweezers.
It's unusual. What else would that be? That's gotta be the eyelid.
And we're supposed to know the fornix?
The fornix?
It's on the lab.
The fornix on the brain, but that does look like
No, the eye.
the fornix on the eye?
Uh-huh. If you
No.
follow... No, we don't need to know that?
Okay.
It's down here.
It's not on there. Yeah, fornix means an arch, is kind of like an arch-shaped structure. There's one that you need to know on the brain, and there'll be one you need to know on the vagina when you get there. Okay, so this is the eyelid, and so this is the ocular conjunctiva. You can see it goes up, and then you can follow it. Makes a little, Bernie said it was a cul-de-sac, right?
Mm-hmm.
Last week. or this week, he does a cul-de-sac, right? Okay, and then, so let's just get a little closer then. Here.
So is the pallbearer all the way to the islet to the cul-de-sac?
Say the question again?
The conjuncta?
The palpebral?
Yeah, yeah, that way.
Yeah, so it's lining the lid, the eyelid.
So to
it's
tip the
this.
eyelid to the cul-de-sac?
Right.
Mm.
Yeah. And so here you can see, it looks like there's some goblet cells in there too. You can see that it's a squamous, or maybe you can stratified squamous. So this is what? What is this little layer called?
Palpebral
Palpebral
conjunctiva.
conjunctiva. Good. That's a mouthful, huh? Okay, we have 10 minutes. Is there anything else with the eye? I think that was it. We can go back to the previous nervous, if you want. Throw up one of those.
Sure.
Throw up one of the nervous system ones for review.
Sounds good.